sí.
Ya
Güey,
está
la próxima vez
transmisión en
creo que hay
vivo.
que, o sea, hay que
Bueno,
mejor
ya,
dar science, que los science son bien pocas figuras.
Ay,
Güey,
ya
pero
sé.
ese se me hizo todavía más fácil que el anterior.
Oigan, creo que ya estamos empezando a transmitir por YouTube, chicos. Creo que ya es tiempo de empezar a
Aceptar, empieza a aceptar.
ya estamos en vivo, amigos.
Voy a compartir mi pantalla.
Vale, sí. ¿Ya lo puedes compartir, Emma, en Facebook?
¿Ya ven mi
Sí.
pantalla?
Sí.
Hay que actualizar ese mapa ahorita que lo veo.
Pues yo les dije, güey, pero ustedes no hacen nada.
Nada, madre.
Máquina.
No, solo hice un video que de nada sirvió. Tanto esfuerzo.
Güey, nadie les dio like, güey.
PJC. PJC.
¿Ya ven mi
Yo les dije
pantalla?
que no hay que hacer videos, la neta, porque nadie. Lo que
Sí,
más
sí,
jala
se ve.
son imágenes, ya nos los dijo Facebook.
Memes de gatos.
Oigan, ¿cómo abro el chat del chat?
Ya aceptó gente, me está saliendo las tres de fuera, pero no sé si aceptarlos.
Ya acéptalos ya.
¿Y Víctor?
No sé, güey.
Oye, es verdad, Víctor no, tampoco se unió en la pasada, ¿o sí? Ah, no, Víctor sí, el que no se unió fue
Güey,
Kevin.
Kevin.
Now live en YouTube.
Hola, ¿qué tal? ¿La reunión no empezó? ¿O
Oh, we're waiting still.
ok, sorry.
Ujú.
Eugenio, compartimos el live en Twitter.
Sí.
Empezamos a las 9 en punto.
Pues no sé, es que si nadie se une. ¿Te gustó el paper, Sven?
No tuve la oportunidad de leerlo, estaba enfermo,
Oh, ya.
pero lo estoy leyendo ahorita mismo.
Ah, ok.
Pero, o sea, de lo que ya he podido leer un poco así en el abstract, es súper interesante. O sea, de que las glándulas mamarias y todo lo que tú puedes transferir al hijo de tal forma, o sea, microbióticamente es muy interesante. Um, yo había trabajado en, ¿cómo se llama? Novo Science Berlin, y justamente ellos tratan de hacer todo también así probióticamente y así mejorar el microbioma y todo, ¿no? Y lo vi interesante y decía, ¿y yo cómo me enteré tan tarde de eso? ¿Por qué me tenía que enfermar? Pero bueno, ya, ¿qué se va a hacer? Ahora tengo que leerlo y ya está.
Okay, so... Sí. Okay.
Okay.
So we're gonna start in a couple of minutes, guys. So remember that every week we have a session of journal club. So if you guys are interested on giving a paper, or you are you want to share some good research, please. We invite you to collaborate with us and to be part of this community of Public Journal Club. So we're gonna start in a couple of minutes. Okay.
So, would you like to start now?
Yes, I
So-
think we could start at least
Okay.
for
So,
the introduction.
in order to save time, I will to introduce the current members of the Public Journal Club team. And today we will present the paper entitled An Immunologic Mode of Multigenerational Transmission Covers Gut Treg Setpoint. So this paper will be presented by Eugenio Contreras, who is a current PhD student at the Autonomous University of Mexico, and the other members of the Public Journal Club team is Emmanuel Flores Espinosa, an undergrad student from Mexico also, and Victor Nieto, undergrad student. Emmanuel studied biomedicine.
Basic biomedical research.
Okay, and Victor studied
Genomical science. I think.
genomical science, also at the Autonomous University of Mexico, and Diego, who is currently a lab technician from Albert Einstein College of Medicine, and Carlos Alfonso, who is a PhD student from Max Planck for immunology and epigenetics on German. And Diego and I both are nearly to start our PhD. Diego on September and me on July if the current sanitary situation let us. So we are the team of Public Journal Club. I hope we hope you enjoyed every session, and also we would like to encourage you to join us on every session, and also to present an paper. And well, so in this session, Dr. Dipshrika Raman will join us at the end of the session to a Q&A round of in this part of the session, I would like to remember you that introduce yourself with a brief sentence. And also, if you have any doubt or you want to ask something during the session, just raise your hand, and we give you the opportunity to talk. And also, if you are shy, also you can ask through this chat, and we ask to Dipshira or Eugenio the question do you have. So that's all. Eugenio, do you want to start?
Yes, sure. Well, thank you guys very much for joining us to another session of Public Journal Club. And today we're going to discuss a really interesting paper entitled An Immunological Mode of Multigenerational Transmission Governs a Good Treg Setpoint. As Aranzazu said, this is a work done by Deepshika Ramnanan, which is the first author of this paper and will join us at the end of the session. This work was done at the laboratory of Christophe Benoit at Harvard Medical School. And this work is entitled An Immunological Mode of Multigenerational Transmission Governs a Good Treg Setpoint. Just before we start, I want to invite you to, if you want to ask or add some comment during during the journal, please hesitate you to create a discussion of this paper. Okay. So, sorry. As a matter of introduction, we know that all immunologists are interested in studying and understanding human diseases. And when we look at the factors that governs or drives human diseases, researchers have found different factors, including different polymorphisms associated with the mutations in genes related with the immune response. We also found different epigenetic factors that drives some factors of disease. In these environmental factors, we can find out nutrition factors that will be related with the development of disease. And now more and more research associate microbiome with the role and development of human diseases. However, and this is really interesting, even though we look for all these factors, there are something missing during this in this puzzle, and in this case, I'm sorry. Okay, there are some immunological traits missing during this looking for human disease relationship, and sorry. Okay, so in this paper, there's a really important notion that there's an Entero mammary axis that actually will program the babies or the progeny for the development or associated of the disease. We know that in this Entero mammary axis, breastfeeding is really important for giving some signals to the newborn, including growth factors, vitamins, microbiota. There are also studies that are associated with the transmission of vertical transmission of immune cells to the newborn. And particularly here, it's really relevant to the role of the vertical transmission of antibodies for the development of a correct immune system in a newborn. So one of the main cells that are studied in this work are the ROR gamma positive Tregs, and these Tregs are located in the intestine. Particular Tregs are well distributed in different tissues, but in the intestine we can find two types of Tregs: the ones that express Helios or FOXP3 Helios positives that are derived from the thymus. And we have an enrichment of FOXP3 ROR gamma positive T cells that are derived, are differentiating after birth, which are called peripheral induced Tregs. And these Tregs are the major subset in the colon, and they differentiate locally in response to bacterial antigens 50 to 20 days after birth. So what are the relevance or the importance of these T cells during intestinal homeostasis? Well, it is well known that these T cells participate in food tolerance and also to control commensal bacteria in the intestine. And using experimental models, when we block or eliminate these T cells, these ROR gamma positive Treg cells, we found that these animals generate microbial dysbiosis and have an enhanced inflammatory Th17 response and are more susceptible to colitis. So, you know, it's really hard to, one of the main question of this work, and it was, for me, was really interesting while reading the paper, because we don't found, or there is no actual initial observation of the whole discovery that they found. In fact, for me, one of the key questions of the whole paper, or how this research started, is which is the mechanism that controls ROR gamma Tregs in the intestine. So I don't know, guys, if you want to add some comments to this. Okay. So just before we start to see all this process, the first thing that they did was that they compare the frequency of Tregs from two different mouse strains, the B6 or the bulb C mice. And when they look in the intestine for the frequency of Tregs, they found that there was actually no difference between the FOXP3 positive T cells in the intestine. However, when they go deeper into searching any difference between the phenotype of these Tregs, they found that the B6 mice have an enrichment of ROR gamma positive Tregs in comparison with the bulb C mice, which have lower ROR gamma Tregs and now have more of Elios positive Tregs in comparison with the B6 mice. This is now called, and I'm going to start called from now a Treg Setpoint, where we see that the B6 mice have more Treg Setpoint of ROR gamma positive T cells in the intestine. And this Treg Setpoint actually is different between the strain of mice. So we can see B6, NOD, or bulb C or CVA, they have different Treg Setpoint between mouse strain. So I guess one of the question was, well, maybe this difference is because of maybe microbiota, and by doing this experiment where having germ-free mice, we know that germ-free mice have a reduced Treg Setpoint, but if we add a specific strain of bacteria that will induce the generation of ROR gamma positive Tregs, we see that by adding Clostridium ramosum or Bacteroides theta, which are bacteria that induce ROR gamma positive Tregs, you have an enrichment of these ROR gamma positive Tregs, but this enrichment is strain or mouse strain specific because of the bulb. The bulb cannot reach the Treg Setpoint compared with the B6 mice. So, and this is the negative control. So it seems that there something in the mice that will limit the generation or the development of this Treg Setpoint at the intestine. Okay. So if this is not dependent by the microbiota, one of the things is that this is maybe driven by genetic factors. So they really did this simple and smart experiment where they interbreed bulb C mice mother with a B6 mice with a bulb C mice, and vice versa, crossing bulb C mother with a B6 father. And when they look at the progeny, this is really interesting to see that the Treg Setpoint is actually inherited from the mother. So in this case, all these F1 generation animals will have a Treg Setpoint that resembles the mother. And this is independently of the strain mice. So we see that here, all these F1 from this cross are enriched with ROR gamma positive Tregs in comparison with these F1 animals that are, which mothers are ROR gamma Treg low animals. So it seems that this Treg Setpoint is inherited or vertically transmitted from the mother to the pup or to the animals. And we see that this Treg Setpoint actually is maintained during adulthood independently of co-housing. And well, this is strain specific or animal specific. This Treg setpoint, which is really interesting, is that it's inherited through many generations. Even if you backcross the animal with B6, or in this case with a Balsi mice, independently of which of the genotype of the father, you will still have inherited this Treg setpoint, particularly from the mother. And this is really interesting. Actually, this is somehow inherited throughout many generations, although I want to make emphasis in something that somehow the phenotype is diluted over time. So we see here now in 1, 2, 3, 4th or 5th back crossing, we see that we now have a down level of the Treg setpoint in comparison with the F1 or the mother. This also happened with the Balsi mice. So until now, we see that there's a Treg setpoint in the animal that is vertically transmitted and is inherited through the mother, and it actually is independent of microbiota, and also it's somehow independent of the genes. I don't know, guys, if you want to comment something about all these results.
So there's a question in the chat. I don't know if you could read it.
Okay.
So, what makes you think is independent of microbiota? Say,
Okay.
Carl.
Well, until now, so these figures, the only thing that is, it seems that it's independent of let's say, microbial strains, is because of this image. Later on,
I would like to say something in that point, that these bacterial communities are able to induce ROR gamma Tregs, but there are no part of commensal microbiota from mice, isn't it?
yes,
So,
yes, because these are germ-free mice that are colonized with a one only strain of bacteria.
Yeah, I guess at this point you can say that it's independent from microbiota stimuli, but you can say it's necessary to, it's required another signals that induce bacterial infection to have this cell population.
Yes, it's somehow tricky of when analyzing this data, and further during the paper we will see that there are many factors that actually induce this Treg setpoint, and it's not only because of the microbiota or the genes. So yes, it's really a good point of observation.
Yeah. So I would like to also add that to really make a difference between the generation of this population and the levels of this population.
Yes.
So in the previous graph you showed, like it's clear that for the generation of the population, the microbiota is required, but for the levels are the thing that they try to say it's microbiota independent. Yes, the Treg setpoint actually here is important. The Treg setpoint is somehow independent of the microbiota presence, and it's somehow associated with the phenotype of the animal. We'll further discuss later at this point. Thank you. So here so this is really okay. So we have this. Yes.
Guadalupe has a question.
Okay.
So
it's just a comment that is independent of the microbiota. In the mouse, we are observing the amount of ROR positive Tregs. I'm thinking about the independence on the dependence on the microbiota. The microbiota here in the mice that show different proportions of Tregs, it's not determinate. It is important in another okay, in the mother, but we see that in a couple of images.
Yes, it's really somehow tricky. What not tricky, but it's really interesting. All the findings that we see that there are actually many factors, not only microbiota or phenotype of the animal. Yeah, thank you. So then now they move to do a really clever experiment, which is so if it's all vertically transmitted, well, what would happen if I swap the pup from a B6 to a valve C mice? And they did this vice versa from a valve to a B6. And what is really interesting here, we see here at the B6 that, well, this is the control, but when we put the pup with the foster mother that is valve C, we see that the Treg set point is actually diminished. And in the contrary, on the opposite side, when we put this valve C pup with the foster B6 mother, the proportion of Treg set point is actually inverted. So with this experiment, they actually conclude that there is no epigenomic imprinting, no mitochondrial DNA contribution, or any immunological modifications impaired during development. So the phenotype or the Treg set point is actually driven just by, it's vertically transmitted, but I guess here at this point, we can start talking about lactation. So this is somehow something happening during lactation that it's driving the development of this Treg set point. So this is really a clever for me experiment. And actually, and more importantly, that this is actually, yes?
We have a question. Hi, I'm John from Kansas City. This experiment you show in the figure 2A, it looks like the interaction itself is more relevant, but also genetic is important.
I don't know if...
Yes, I guess this could be important because you can see clearly in the figure 2A that something is enhancing and is repressing in the genetic baggage of the mice. So when you switch the pups to another mother, you can see that the PLC is suppressing the BC phenotype. Maybe it's another interpretation of this figure. It's my interpretation. So when you see in the valve C, you can see also BC is enhancing this ROR gamma Treg cells production or promotion. I don't know. So what do you think, John Pitch, care about that? Or what is your thought about how is important the genetic environment and it can be excluded of this mechanism?
Jesus Benito has a question.
Okay, Benito.
Okay.
Thank you. It's more like a comment, maybe about what John Pitcher is saying, that also the phenotype is changing when this cross fostering. For example, in B6 mice fostered by valve C mother, the set point of Tregs is decreased, but it's not at the level of valve C mice fostered by valve C mother. So the fostering it's important, but as the set point is not the same as the B6 mice fostered by B6 mother, you can say that there's a contribution of both factors.
Yes, you have a really good point. Although we don't have statistics, it seems that there's a difference here. And actually, I'm going to start adding some comments about this. So there's actually some data that in the supplementary data that mentioned that they did some analysis where they contribute that there's a 60% of the genotype is contributed by the foster mother, but there's some 27% of the genetic compound of the pup that will in fact alterate or generate this phenotype. I also want wanted to mention that this fostering, when these animals are fostered, they are this fostering process or the set point of this fostering process is actually inherited, which is really interesting. Now we see that there's no actual somehow genetic contribution, but it's more important actually the phenotype or the set point of the foster mother for this inherited set point generation. So one of okay, so we see that there's a vertical transmission of the set point. So one of the main questions is what are the if there's any window that is important for the generation of this Treg set point. And when making this experiment, they once again they swap the pups from a B6 to a bulb and vice versa. And we see that, for instance, for the B6 phenotype, there's a really small window of time from 0 to 3 days where when you swap the pup, you will reverse or will have the Treg set point from the mother. But if you swap the pup from day 3rd to adulthood, we wouldn't see now this Treg set point. In the other hand, when in the biopsy mother, we still have somehow the same phenotype, but we have a more open window from, I think, from 0 to 7 days of time window where you can swap the pup and you will somehow get the Treg set point. So this is really interesting. So we have a really short period of time that will make this Treg set point difference.
Okay, Eugenio, we have another question from
Okay.
David, Master, Manchester University undergrad in biology. Open question for everyone. What happens during the day 3 to 7 during the development that it's so important to define this phenotype? I would like to answer this question.
Yes.
That they say and they remark to, especially in this figure, that this correlate with the recovery of microbiota. And I don't know if you also think, Eugenio, that the set point in the establishment of microbiota is different of these strains of mice, and this maybe is changing the set point of ROR gamma Treg through the development, because as you can see in the first graphic, in the first plot, you can see the B6 strain looks to be more maintains this population through the time than the population.
Yes, I don't know.
One important feature of this figure, sorry, Eugenio, is that also it's important to mention that ROR gamma positive Tregs are absent in the mice until 2nd week after birth. So in this day, like 3 or 7, there is no ROR gamma Treg cells in the colon. So it's interesting how these processes are imprinted so early, even before that these cells are present in this tissue. So also, what is about the question? What is happening in 3rd and 7th day after birth? I don't know, but there is interesting that in the mice from biopsy that are fostered to B6, there is a more effect at 3rd day that is absent in the opposite change. So I don't know if someone have any comment about this effect.
No, I don't think there's a really open question here. Actually, Victor asked, what's the difference between, why have bulb C have more open window. I don't know. It's maybe perhaps because of the
Strain.
or the IgA producing cells that we further will see during the figure. I don't know, but it's really a good point that during this time in this first stage we don't have any Treg at all. So
Yeah. Okay.
so when looking at what is the physiological relevance of having or not having Treg, and what would happen if we foster these animals with a B6 or a bulb C, when infecting this animal with Citrobacter rodentium, which is a bacteria that will induce intestinal inflammation, we see that when measuring after the period of infection, I think it's 2 weeks after. Let me check. It's 2 weeks after infection, we see that shortening of the intestine, which is associated with the inflammation. And we also hear in the bacterial the bacterial load at the stool, we see that actually the bulb C is able to eliminate the bacteria in compare with the B6 animal or B6 foster animal, which indicates somehow that there are more Tregs that are actually inhibiting inflammation. And we confirm that when analyzing IL-17 or interferon gamma, that the bulb C animal are more prone to produce these pro-inflammatory cytokines compared with the B6 fostered animal. So we see clearly that there are some physiological results of these Treg Setpoint in different inflammatory conditions. And with here, they make this really interesting experiment. Yes?
I would like to also say that next to the plots, the dot plots.
Yes.
There is a figure about the correlation between IFN gamma and IL-17 production, and I would like to say that it's pretty interesting how the bulb C strain looks to be less inflammatory than the BC. So yeah, and the plot of the correlation between the interferon gamma and IL. Yeah. You remember that?
Yes.
Yeah.
Yes.
So it's interesting how the bulb C foster looks to be less proper to inflammation than the
well, I think in fact the bulb C have more inflammatory producing cells, but at the end of the 2 weeks of the measurement, they have actually less inflammation. We see it here in the intestine with the intestine length, and this is maybe because I'm guessing because of that they're actually controlling the infection, and now we don't see we see less bacteria, which now correlates with less inflammation. So I think that's my guess, and this is all because we have less Treg. So we have a question from Joaquin Hidalgo.
Hi, Joaquin, from the University of Valencia. ROR gamma Treg cells positive are protective against colitis. Why do the mice with higher levels of ROR gamma Tregs develop more severe colitis?
Levels of ROR gamma Treg develop more? No, actually, because of in this, at least in this model, they suffer more inflammation because they are not controlling they're not controlling the bacterial loads. So you have a persist inflammation. Yes. So I think I'm gonna move on a little bit faster. We have less than 30 minutes. So when in this case, one of the things that they made, I'm sorry, is that maybe this Treg Setpoint is determined or associated with the Treg population. So what they did is having this diphtheria toxin animal system where they can deplete Treg. So when they deplete Tregs with giving the diphtheria toxin, we see that, okay, the system actually works. We don't see any Treg, but after 4 weeks of recovery, independently of the elimination of the Tregs, you will still have this, you will still reach this Treg set point that is somehow inherited from the fostered animal. Even if you are a B6, you will have more Tregs. If you are a BALC, you have less Tregs. So this is really quite interesting. That is, this Treg set point is independently of the presence of Tregs. And here, more important, that when giving antibiotics, okay, so maybe this Treg set point is somehow associated or maintained with the presence of microbiota. So in the adult animals, they treat the animals with the antibiotics. They wait 4 weeks to recovery and analyze now the Treg set population, and we see quite interestingly that even if the animal is recovered after the depletion of the microbiota, you will still reach the Treg set point that is inherited by the mother. So in this case, this is the control, the B6 foster with a B6, the B6 foster with a BALF. We see a recovery of the Treg set point. This is also happening with the BALC foster with the B6. So the same conclusion here, that independently of the presence of the bacteria, you will have you will reach this Treg set point that is once again inherited by the mother. And here I think it is really particular and really interesting figure of the paper. What happened during inflammatory conditions? In the last figure, in this one, we see an inflammatory condition associated with a pathogen. In this case, they are generating an sterile inflammation in which we are destroying now the tissue, giving DSS. So this is by giving DSS, you will somehow erase the Treg set point that was inherited by the mother, and independently now from which animal you were fostered, you will reach this Treg set point equally. And more important here, that this Treg set point is actually inherited. We'll see it. I'm sorry. We see it here. So this is the control with no inflammation. This is the animal that is that are induced inflammation with the DSS, and now we see that this Treg set point is actually inherited. So what is important of this figure is that it's not the microbiota, it's not the genetic strain of the animal. It's what is actually inherited from mother. What is actually vertically transmitted from the mother to the pup is the Treg set point. In this case, independently of inflammation. So we have a question from Sophie Helm.
Sophie Helm. Hi, Sophie from University of Poznan in Poland. In the DSS phenotype resistant to other perturbations, Tregs or microbiota depletion?
Is the DSS phenotype resistant to other perturbations? So the question is, if you deplete, well, if you did you deplete Treg? Well, you don't have. I'm not guessing this question. I mean,
if I can understand. So, is the DSS phenotype resistant to other perturbations? Tregs, microbiota. If I understand well, your question is that you can see an additive phenotype when you see the DCS phenotype, how microbiota depletion could shape in this phenotype. Is that Sophie help?
I don't know.
Okay. Well,
I will go on.
While Sophie think about that, I would like to say that, so if you if mice transmit this set point of inflammation, So what is the point to reset the inflammation set point in every generation? So make you less immunogenic to foreign microbiomes, or what is the point to
yeah, what is the point of inheriting the Treg setpoint? Well, I think I will try to think more in evolutionary way, that maybe somehow the mother will try to inherit this Treg setpoint that will protect their progeny, assuming that the progeny will have to deal with the same inflammatory cues as the mother, so it's somehow protecting.
Yeah. I understand that, but also when you are more inflammation, you also have to develop autoimmunology disease.
Yes.
So there could be a balance between those points, that you have to be able to respond against pathogens, but also you have to be able to maintain some homeostasis system. So because that also is favoring the progeny.
Yes.
During the generations.
Yeah, it's really a good point, and there's many things that we can explore of this paper. I think it's really interesting. So I will move on. We only have, I think, 15 minutes. So we also know that I put here some of the results of the paper, that this phenotype is independent of the tissue repertoire. So there's not any contribution of fostering difference between cross-fostering in tissue repertoire, and there is also no contribution if you co-house the animals at birth or later during after weaning. So there's no actually contribution of weaning of co-housing. What is really important here, that okay, we think that and the main guess is that it's something that is transmitted in the milk, or maybe in the microbiota on the stool, that is actually resulting on this phenotype. And what they tried to do, but they were not locally, they didn't have any results about this, is that they give milk gavage or stool gavage instead of cross-fostering. They are giving milk from a bulb or stool of a bulb or a B6, and they were giving the pups 3 days during 3 days after birth for only 3 days, and they didn't find any difference at all. This is really not conclusive, because of these animals were also giving they were with the mother, with actual mother, so we cannot separate the results of giving milk or stool gavage from other phenotype with the association that they were actually with the mother the whole time. So this is not, sadly, this is not really a conclusive experiment at all. And this is not conclusive because of it's very difficult, actually, these results. So, okay, so maybe, and we have really here some contribution that maybe it's because of the microbiota. So we have this clue that maybe perhaps is the microbiota. So what they did is that we have the germ-free mice, which we know that have less ROR gamma Treg setpoint, and they transfer microbiota from a stool from different bacterial, or from different mice strains. So giving B6 or TBA or BALC, putting into a germ-free mice. And what is really important here is that these treatments, they reach the same Treg setpoint. So the Treg setpoint is actually independent of the type of microbiota that could be in these different bacterial strains. This is really, really important for understanding the whole figure of the paper. And they did some analysis trying to find any difference at all in the bacterial strains. They don't find any difference between strains, or if the only difference they found, they were only between cages, or any difference between the pups that were fostered with any other mother. So there was really not conclusive results in these figures. There were actually no difference. And just to resume some figures, they mentioned that B6 or BALC mothers do preferentially pass on different microbiota, particularly Clostridiales, to the offspring, but this appears unrelated to the Treg setpoint. So the Treg setpoint, once again, is independently of the type of microbiota that the mice or the animal have. What is really important, what is important, what is the importance of the microbiota? The importance of the microbiota is in the mother. So in this experiment, what they did, they give they give antibiotics 5 to 2 days, the last 5 to 2 days of gestation before cross fostering. So in this case, the bulb pups will be fostered by a B6 and vice versa. And we see particularly here that the B6 bulb C animals that were fostered with a B6 mother that have here a depletion of microbiota, the last day of gestation, we see that we see a loss of this Treg setpoint inheritance, which is really important. So the microbiota is only important in the last day of gestation. Let's say this is really my guess. I don't know, guys, if I have any point of this, but. And this is
Lens has a question or comment. Sorry.
Yes, hi. I was just really wondering about this figure because, like in 1B, they show with the germ-free mice that there is still a difference between black 6 and bulb C. So I was really wondering that this few days of antibiotic make that difference when they black 6 and the bulb C germ-free maintain their level of Wargamma T positive Tregs.
Yes, I think it's a really good question, because my guess is that maybe you have this little time window before birth, or that maybe will induce the migration. The presence of the microbiota will induce the migration of the IgA plasmablast. I don't know. It's really a good point, because it's somehow confusing why you require the microbiota just before. They don't show any data of what's happening with the IgA plasmablast in the breast, in the breast, or any at all. What's that? I don't know if it's that somehow answered your question.
I think in part, but thank you.
Yes, but I mean, luckily we will have the first author, so I think it's a good opportunity to ask all these good questions to the author. So, okay. So they see all this phenotype, and then now they're trying to look if there's any difference at all on T cell activation marks. And what they found is that the bulb C have less T cell activation compared with the B6, and there are actually no difference between the Treg activation state between strains, independently of the expression of ROR or alleles. What is really, yes?
We have a question on the chat. How germ-free B6 versus bulb C maintain the ROR-gamma Tregs levels while this short antibiotic treatment before birth change it's so dramatically.
Before change.
Sorry, can I just comment on this? This was from me just because someone else could not hear the question. It was the same that I asked before.
Okay. Yes. Thank you, Verena. Yes, I think it's really an unsolved question. I don't have any clue of that. What is I think we are not making this this story where when we look at any other difference, they found really here an important difference between the the IgA producing plasmablast cells. So we see that in the colon lamina propria, the BALB/c mice have enriched IgA producing cells and compare with the B6 mice. So maybe this could be a trait or a striking difference that could lead to all these difference that we have seen through the paper. So one important thing is that these animals produce less, the B6 actually per se produce less IgA in compare with the BALB/c. And this trait is actually inherited through many generations. So the BALB/c produce more IgA, whether in the serum or in the stool, or we can find in the stool of the animal. So, and what is more importantly is that what the main function of IgA, it's in fact the protection or the control of microbiota. And when we analyze the IgA-coated bacteria, we found that the BALB/c actually have more IgA-coated bacteria in compare with the B6 mice, and we found a negatively correlation between the presence of IgA-coated bacteria and ROR+ alleles Tregs. So in this case, the B6 have more ROR gamma positive alleles Tregs and less IgA-coated bacteria. And this immune trait is actually inherited during this experiment of cross-fostering when, this is in adulthood, this is in the neonates when we see this phenotype, and when we cross-foster the animals, we see a this same trait where the BALB/c, this is the B6 globe. The BALB/c animal fostered by a B6 have a less IgA-coated bacteria. Actually, it's mostly the same as a B6 foster, and this this IgA-coated bacteria also phenotype also is inherited when B6 is fostered by a BALB/c. So another thing that is really now important for the story that are telling us that actually when the authors quantify the IgA in the milk, we see that actually the BALB/c produce more milk. We have more milk. We have more IgA in the milk in the BALB/c mice compared with the B6 mice. And just to mention that this IgA-coated bacteria phenotype is actually inheritable during many generations. I think, yes, I'm sorry. So what's going on with the IgA? So they found the authors that the IgA plasma cells are actually enriched during the last stage of gestation, and during lactancy, we see enrichment in the colon, in the small intestine, and actually in the mammary gland. So they did this really smart experiment where they we found IgA in the plasma in the mammary glands, but we don't know where this IgA come from. So they used this incredible, really interesting animal mouse model where they can actually photoconvert cells from any anatomical tissue and later go on to see if there are actually any migration at all. So they photoconvert the animal in the intestine, and they found days later that we have red cells now in the mammary glands. So here is the graph where we see that after 2 days of after photoconversion, we see that there are some migrations from the intestine to the mammary glands. So this migration is not only associated in the mammary gland, but you also have migration in the spleen, I think, and also in the bone marrow. This is really interesting. And for me, this last figure is also interesting because maybe the difference or the striking difference that we see is associated because of we have, I don't know, maybe less IgA producing cells in these both strains. And importantly here, what you see that in the mammary gland, independently of the strain, you have the same numbers of IgA producing cells. What is in fact given the difference is that the amount of IgA in the milk in the bulb C compared with a B6. So this is I think a good result, because the difference now is in the amount of total IgA in the animal. So it's really quite interesting. So we have at this point we have this model where we know that and it really like somehow a cycle or a negative loop. We have the adult that in the mammary glands they will produce IgA, and we'll give it to the will vertically transmit it to the neonates, and this IgA will coat bacteria, and this IgA coating will negatively regulate ROR gamma positive Tregs, and this somehow will negatively regulate IgA B cells. So it's a double negative feedback loop. So the question is, well, do the ROR gamma Tregs inhibit IgA plasma cells, or the reverse, or both? So they did some last experiments where they had these B6 mice and they fostered with a bulb C, or with a bulb C deficient of antibodies, deficient of the G chain, and we see that not having if the foster mother don't have antibodies, the phenotype resembles as a B6. So we have more ROR gamma positive and less IgA coated bacteria, and this is not happening with the bulb C. And here is now this is co-fostering. This is actually cross from a B6 to a B6 or above C deficient of G chain, and we see that in fact the here we see that there's a really important factor that the mother needs or require the bulb C to the G chain to inherit this immune Treg setpoint. And lastly, well, this is, I think, this is the graph. Yes, not having IgA or not having IgA will resemble as a B6 if you are a bulb C. And finally, yes, finally, if you have an, well, okay, so this is somehow dependent of IgA producing cells or the presence of IgA. What would happen if we don't have Tregs? If this inheritable strain set point or phenotype is inherited, so now we have this animal that we deplete ROR C in all the FOXP3 pool, and not having not having Treg in the this ROR positive Treg in the intestine will in fact repercute or have a direct impact in the IgA coating bacteria frequency in the animals. And it's really important here that when depleting alleles is not affecting the phenotype. So it's somehow dependent. So with this experiment, they conclude that it is dependent. The phenotype that they observe is dependent of IgA producing cells and somehow dependent of IgA coated bacteria. So we have this model.
Can I comment something?
Yes.
So in this, for me, it's interesting that although they deplete IgA or they deplete the ROR C, like, you still have a certain level of, well, in depleting IgA, you still reach a certain level of ROR gamma positive Tregs. So it's not like the level just skyrocket. So there's another thing that it's not the IgA that is controlling the levels of this cell population.
Yes, I think there are many factors that governs this phenotype, and there are a lot of work that they could do. I don't know if anyone has any other comment about this this work.
Yeah, I think it's pretty interesting how well, when in the figure 3E, you have the core microbiota contribution through the different mice strain. So I was wondering if, well, lactobacillus is are a species that are less immunogenic than other bacterial communities. So I was wondering if ROR gamma Tregs have a crucial role in maintaining homeostasis at the mucosal barrier. If this trade-off between homeostasis signal between communities associated with tolerance and ROR gamma. So you can have both tolerance signal in the gut, because this could be very expensive for the system, and it's bad for and it's bad for maintaining the offsprings, and it's difficult to allocate a lot of resources to these responses. So I don't know, what do you think?
I'm sorry. Yeah, I mean, with the thing of the microbiota, I'm having a lot of issues or controversy, because because of the fact that in the germ-free or giving in the germ-free mice, when you give a stool of any of different strain, you will reach this Treg Setpoint. But I don't know. I don't know if anyone in the public have any
So okay,
jump picture.
join us, Dipshira. So
Okay.
well, guys, I'm trying to introduce Dr. Dipshika Raman, the first author of the paper we have just discussed. And she's identifying function of regulatory T cell subsets and the intestinal inflammation at the Dr. Diane Meredith and Dr. respectively, and as a producer researcher at the Harvard Medical School at Boston. And during her PhD, she was understanding the mechanisms of NOT2 microbiome interaction in intestinal disease at the Dr. Kev Cadwell lab at the New York University. And she's a molecular cell biologist focusing on the immunobiology and inflammation process. Dipshira, we are excited to you have been here today with us on the Public Journal Club. So welcome.
Hello, thank you all for having me.
So once again, I kindly ask you to introduce yourself in a brief sentence. And before make your welcome, if you are shy, don't worry. We can read your questions to Dipshika. Just ask through the chat. So we have our first question on the chat. Are IgA levels change in different organs related to dietary change in pregnancy?
Good question. So we haven't looked anywhere except the gut. I'm guessing you're wanting to know if it's different in the lung or other organs like that. Is that the question?
So Mark Bayer. Hi, postdoctoral fellow at Frankfurt University. I work with innate immunity in flies. If ROR gamma Treg cells are protective against colitis, why do the mice with higher levels of ROR gamma Tregs develop more severe colitis?
I'm sorry. So can I just repeat the question?
So also you can read through the chat.
Oh.
So sorry. Mark Bayer asked, if ROR gamma Treg cells are protective against colitis, why do the mice with higher levels of ROR gamma Tregs develop more severe colitis?
So this is an interesting question. So we tried two different types of colitis. So here this is the Citrobacter rodentium colitis, right? So it's an infection-mediated colitis. So with the infection, what we found is that, like, as you can see, mice that have higher levels of ROR gamma Tregs get more inflammation. And we think the reason for that is because the ROR gamma Tregs are there to sort of suppress the increased IL-17 or IL-22 you would get in response to several commensal microbes in addition to, pathogenic microbes. So this is sort of like two sides of a coin. You need, to respond against, autoimmunity, but you also need to respond against infection.
Mhm.
So we think the reason why the mice with more RR gamma Tregs are getting more colitis with Citrobacter is because they're not able to mount as good of an IL-17 or IL-22 response, because you need those cytokines to dampen Citrobacter colonization. Does that make sense? So basically, when you have more RR gamma Tregs, you have less IL-17 and less IL-22. So you end up getting more inflammation in that context.
Okay.
So hi, Deepshika. Thank you very much for accepting our invitation in our public journal club. I have a question related actually to the last figures where you showed that here. Let's see. Yes, here. That actually there are no difference between the frequency of migrating cells towards the mammary gland, but it's in fact the amount of IgA that producing bulb C. So what is there any factor that is driving that bulb C plasma cells produce more IgA in compare with B6?
Yeah, so we tried to do that. So I tried to, isolate the B cells from the mammary gland, and I tried to do an in vitro assay to see if I would see more IgA, but it's just so hard because, the level of IgA I was getting was not detectable by ELISA. So our theory is that, the amount of IgA is not just increasing in the mammary gland, that it's increasing systemically, right? Because even in the serum, we do see more IgA. So we think that the IgA that's coming into the milk is not just the IgA from the mammary gland, but it's also the systemic IgA that's being transported. So one thing we wanted to see is maybe there are differences in, the receptor, like PIGR receptor or other receptors that would, transport IgA into the milk. That we have not tested yet, but we want to test that to see if it's maybe the transport of IgA that's different between the two strains, or if it's just as simple as, well, there's just more total IgA in the serum too, and that IgA is being transported into the milk.
So what happen if you inhibit the migration of IgA plasma cells to the mammary glands? You will still have the same phenotype?
That's a really good question. So we haven't tried that. So something we looked into is you can use CCL28. You can block CCL28 and you will no longer get migration of the IgA positive plasma cells to the mammary gland. But we have not tried doing those injections. Yes, but definitely we could try that.
Okay.
Thank you.
Thank you.
Luis Angel asked, if we consider that T cells could change its function depending on the cytokines on the microenvironment, these 4 gamma T cells could be an effect of transdifferentiation and could exist other kind of Tregs with GATA3, Tibet, or something like that?
Oh, I see the question over there too. So we do find in the gut, it's not just, so here we just look at the difference between RR gamma Tregs and Helios Tregs, right? But you're right in that in the gut, there are also GATA3 positive Tregs, there are also Tibet positive Tregs. And we do see the proportions changing of these Tregs as well. So yes, at any given time, you have all of these Tregs. Your question about can you change it depending on what cytokines there are, so far with everything we've tried in adult mice, the proportions don't really change. Unless we do something severe like DSS colitis, no matter what you change, the proportion of these Tregs don't change. But it's possible that when the pups are neonates, right, maybe changes when they're neonates in the cytokine levels could affect whether you're going to have more RR gamma Tregs or more GATA3 Tregs and so on. That we haven't tested. So we could try injecting neonatal mice with different cytokines and see if we see a difference there. But we have not tried that.
Okay, So,
Aranza, I think there's a question from Verena.
okay. So, Elena?
Verena from.
Verena. Yeah. First, Sarah Holt. I'm curious if the DSS phenotype resistance to other perturbations, Tregs or microbiota depletion.
So you're saying give DSS to mice that no longer have Tregs or antibiotic treated mice? Is that the question?
I don't know.
We don't know.
Well, because DSS is pretty, well, giving the DSS is a pretty strong chemical. So these mice already get pretty sick. We haven't tried antibiotics with DSS. I'm assuming the mice will probably die if we did that. But I'm not sure. We haven't tried giving DSS to mice that already have other perturbations, but that's an interesting question.
So
I'm not sure what would happen.
Chica, just following that question, maybe what would happen the opposite way? Like when you put DSS, you get like higher levels of gamma and T. So when if you deplete those high levels, what happens afterwards? Like you get again high levels, or you like restore the phenotype you had before DSS, even before DSS?
So it's really, so you're saying to basically, once you have DSS treated mice, to deplete Tregs after they have DSS. Is that you're asking? So we haven't done that because DT treatment is pretty toxic, as you can imagine. And DSS is also pretty toxic. But what I can tell you, which we haven't published here, but we have done in the lab, is if you have mice that are deficient in RR gamma Tregs, so that have no RR gamma Tregs at all from birth, so the conditional knockout, if you give those mice DSS, they die pretty quickly compared to their pre-negative controls.
So related to this question is quickly, did you measure the IgA levels in mice where you depleted Tregs, like when you applied the Treg toxin? Because you have depleting the repression of the one mechanism of repression of IgA secretion or expression. So did you analyze if maybe removing this negative expression over IgA expression could induce the rising in the IgA level?
Yeah, that's a good question. So when we did the DT treatment, so you're asking well, I don't have the figure here, but in the DT treated mice, if we just looked at the IgA levels in the stool and serum, that's what you're asking, right?
Yeah, yes.
Yeah, so yeah, exactly. So that figure. So unfortunately, when we did this with DT, we didn't see any change. So when we just give DT, when the Tregs are completely depleted, right, we don't see any change, but that's it's only a week, right? So maybe that's not enough time to see a change. But when we see this recovery over here, I'm using my mice as though you can see it, but basically plus DT, where you see the three mice, the controls to show that the Tregs are depleted, we did not see a difference there in IgA. But the one right next to it, where it's the plus DT plus the recovery of the Tregs, there we do see a difference in IgA. So mice that basically come back as having low RR gamma Tregs did have high IgA, and vice versa. So mice that did have high RR gamma Tregs, they did have low IgA. The recovery was also of the IgA, not just the RR gamma Tregs.
I'm sorry, just one more comment regarding to all these questions. So in this case, in the DSS animal, what happened with the IgE producing cells? The IgE amount in these animals is the same or not?
No. So IgA, when we give the mice DSS, it was sort of all over the place, but that's why we didn't include it on here. We're guessing it's because, you're disrupting the epithelium so much, everything is changing so much with the DSS treatment that the levels of IgA we were seeing is also a result of that.
So once again, in both animals, you will still have the same amount of IgA, and that will lead to the phenotype in the progeny.
Oh, I see. I see. You're asking about IgA in the progeny of the mice that got.
Yes.
Yeah, we have not checked that. What we did check is in the panel on top, where we give the mice DSS. We checked the IgA levels there, but we did not check in the pups. But
is there any difference in IgA here in H?
No, all the mice are the same. Not same, but very similar in IgA.
Okay.
Okay, and Barena asked, in the line with Eugenius' question, have you tried giving the pups with pure IgA instead of milk?
That's a really good question. So we did consider that. We just, buying pure IgA is extremely expensive, and we would have to buy a lot to give it to the pups. And so we sort of didn't do that. One thing we considered doing is getting from the milk of the pups to kind of cross-absorb IgG and IgM and all of that, and then try to give the milk. So technically then the milk would just have IgA and not the other antibodies. But it's not as pure. Yeah. So the answer is no, we haven't done it, but we've considered different ways of doing it that is not so expensive. And yeah, we would like to do that. And another thing we also want to consider doing is, if we say supplement with formula and see if we see something similar, and then we can do formula plus IgA or something like that to see, is it, just IgA that's contributing to this, or are there other factors? Yeah. I can tell you that our hypothesis is that it's not just IgA. We do think there are other factors that are contributing to this as well. But we don't know what those factors are. Yeah.
So I also wanted to mention a previous question from Verena that says, how do germ-free B6 versus biopsy maintain the raw gamma Treg levels while the short antibiotic treatment before birth changed so dramatically for age, I think.
Sorry, it was your voice was cutting off. Could you?
Yes. So how do germ-free B6 versus biopsy animals in 1B? I guess I will move my images for you could. So here.
Yeah. Okay. Yeah.
So maintain the raw gamma Treg levels while the short antibiotic treatment before births change so dramatically.
So these are germ-free mice. So these are completely germ-free mice from birth. So they haven't had any bugs. They're not given any antibiotics or anything.
Okay.
These mice are completely germ-free, and when they're 4 weeks of age, we take them out of the germ-free isolator. So until then, they've been completely germ-free, and at 4 weeks, we take them out of the isolator, and then we gavage them we monocolonize them with one of those bugs. And we colonize them for 2 weeks, and then we look at the phenotype. So they haven't gotten any antibiotics or anything. So they have no RR gamma, not no, but they only have 10% RR gamma Tregs before they're colonized with anything.
Hey, I hope it's Verena, so I hope I can jump in to maybe clarify. So I was wondering about this figure 4H, which Eugenius shows right now, because there you have done this treatment with antibiotics.
Yes.
Just I think 2 to 5 days before birth.
Yep.
And I was really wondering about this difference to the germ-free, because the germ-free seem to maintain, so the black 6 have high raw gamma T and biopsy low. And here, when you treat them with antibiotics, just for some days between birth, it messes it all up.
So those are two different things. So here, we're not treating the pups with antibiotics. We're only treating the moms with antibiotics here.
Yeah.
So here, when we're treating the moms with antibiotics, and we're giving them pups after they've been treated with antibiotics. So we want to see, does the presence of microbes at birth matter? And it does matter. So you need to have microbes at birth to get RR gamma Tregs in the future.
The germ-free also. So it's about the pups having.
It's about the pups having the microbes. Yeah. Yes.
Okay, so the foster mother was not treated with antibiotics, but
So here the mothers are not treated with here the mothers are treated with antibiotics when they're pregnant. And then when the pups are born, we get rid of those pups that were born to that mother, and we give them new pups because we don't want the effect of antibiotics during gestation. Right? So we give them then new pups. So these pups are only getting milk from mothers that were treated with antibiotics.
okay.
And that seems to make a difference. You need to have microbes in those last few days of gestation. The mothers need to have those microbes in order to pass IgA or the other factors to the pups, who will then develop ROR gamma Tregs later on.
But still, the germ-free also don't have that microbes. Like neither at birth nor the mothers that then give milk to the germ-free pups.
So you're right. So in the germ-free mice also, that's why we don't see any ROR gamma Tregs unless we give them a bug later on, right? So what's happening here is, so for example, let's take an example here. So the B6 mouse that was treated with antibiotics, when we give them pups, like biopsy pups, they're all low. They all have low ROR gamma Tregs, right? They don't get as high as B6 normally would pass on. And similarly, in germ-free mice that are, germ-free that don't have any bugs, they're always low ROR gamma Tregs. So without microbes, it's always low ROR gamma Tregs. And then in the B6 background with microbes, you get high ROR gamma Tregs. Does that answer your question?
Maybe I'm not really getting it, but I don't want to kind of interrupt everyone else from asking questions.
We can talk about this again. We can.
Would be cool if there is some more time at the end. Thanks.
Yeah.
Victor from Mexico asks, you mentioned that Ig coding of inducing microbes probably is not the only regulator of the observed phenotype, and that maybe a maternally controlled regulation helps to modulate the influence of the factors that are involved. Have you any idea in which way?
So that's a good question. So we're wondering that ourselves. But I will say that there are several studies that have come out about factors that are regulating ROR gamma Tregs. For example, short chain fatty acids. There are bile acids. There could be cytokines, like someone else mentioned before. Cytokines like IL-6 is really high in the first few days of birth in the milk. So it could be these cytokines. And one other thing is there's a very recent paper that is also talking about how stem cells could be transferred in the milk, that's helping in the development of neurons, but it could be there are other cells that are being transferred that could be useful in the intestinal niche. So it could be any of these factors. There's also this other paper about how milk can affect gaps in goblet cells. And it could be that the goblet cells are being impacted by the mother's milk, and that's how these ROR gamma Tregs are being regulated. So there's several other factors that could be playing a role here. We do want to follow up on some of these to see which of these, in addition to IgA, is causing this phenotype.
Okay. Li Xuan asks if IgA code bacteria inherent via microbiota, could that affect the development of new ROR gamma Tregs?
So new ROR gamma Tregs in the sense of an adult? So you mean if you give, I'm not sure I understand that question.
What Li Xuan asked answer. I was wondering, Dipshira,
yeah.
why biopsy mice harbors high loads of lactobacillus, and if this could be given an interplay of RORY gamma T cells, Treg cells, to establish a tolerance mechanisms. And another question, if RORY gamma Tregs have a crucial role in maintaining the homeostasis at the mucosal barrier, if this is a trade-off between the homeostasis signals in the gut from BC strain and BAPC strain.
Okay, so one quick thing that I'll clarify is, so the microbiota doesn't come coded already, right? You get colonization and you're also getting IgA from the milk, and so they get coded in the gut. So the microbes don't themselves already have coding, if that makes sense. And also in terms of the lactobacillus that you asked in the biopsy, it's tricky because if you look in the graph, we also found B6 pups that had the same lactobacillus,
yeah,
but they had high RR gamma Tregs. But that same lactobacillus in biopsy was causing low RR gamma Tregs.
Mm-hmm.
So the question you're asking is it because the lactobacillus coding of that, I mean, the IgA coding of that lactobacillus is high versus low in those strains, and if that's what's causing the RR gamma Treg phenotype, right?
Yeah, right.
Yeah, so we tried to do that experiment. So we tried to give that lactobacillus to germ-free pups to see if we could see a difference, in coding, but that didn't really work. So it's kind of hard to do that experiment, right? Because the only way for us to do it is to take milk from a high mom and a low mom and give it in addition to lactobacillus, but that's not really germ-free. So we could be introducing other microbes by doing that. So it's sort of technically challenging to show that, but you're right. We do think that it could be that the difference in coding of those bugs, even though they're the same bug, the difference of IgA coding is what's contributing to the RR gamma Tregs.
Yep. My last question is, well, we have a past session, and I just was wondering if how organ-specific isoforms of fatty acids binding proteins could be shaping this phenotype, these different strains.
Yeah, that's a good question. We don't know,
Okay.
but that's something that we do want to look at. We do want to try doing single cell RNA-seq. We do want to do RNA-seq because we haven't done those sorts of experiments here in this paper. So that's in our future directions to do that, to see if we can find any differences there in the intestinal environment in terms of these receptors.
Okay, thank you so much. And well, each one say in pups, his question was regarding that in pups, in mice pups.
The first mother gives milk, but pups are also in contact with its microbiota. Yeah, so that's what, so here the question said, the foster mother gives milk, but pups are also in contact with its microbiota that could potentially affect RR gamma T cells in the pups. So that's also one of the reasons why we think the composition of the microbes doesn't really matter. It's more the coding of the microbes that matter, because you're getting different microbes from your birth mother, and then you're also getting these microbes from the foster mother. But for whatever reason, you're always following the foster mother's phenotype if it's in that narrow window within the first 3 to 7 days, right? So we think that it's because the IgA you're getting from the foster mothers, or IgA plus these other factors that you're getting from the foster mothers, is different because the microbes are different from both the mothers, but yet you're always consistently getting either a high or a low phenotype. So that's why we think the type of microbes are not really playing a role here. It's more their coding that's affecting it.
Okay.
Guadalupe raised the hand.
Okay, Guadalupe, go ahead.
Hi, Dipshika. I'm a PhD student from the National Autonomous University of Mexico, and I have 2 questions. The first one is if you think that is it important, the specificity of IgA in the induction of this Tregs? And the second one is if do you mention that it's not that is a little contribution of some genetic factors? Do you have any idea which factors could be these that are modulating the proportion of the Tregs? Because for me, it is difficult to think about a genetic factor that could modulate the amount or the frequency of those Tregs.
Yeah, so great questions. Again, questions that we're wondering ourselves also. So in terms of the genetic thing, if you look in our data, you will see that, for example, B6 mice are anywhere between 40 to 60% RR gamma Tregs. And if you look at biopsy, they're anywhere from 15 to 25-ish%. But when we do the cross posturing, or even in the F1s, we found that there's a threshold above which the opposite mice don't go. So for example, biopsy mice that were fostered by B6 mother never went up above 40%. And B6 pups that were, fostered by a biopsy mother never went down below 30%. So the only difference there then is, either genetic or, some sort of epigenetic factor that's playing a role there. And what factors they are, we have no idea. But one thing we do want to do is, if you notice, we did do these back crosses for several generations. So we sort of want to follow that and sequence the mice so we can try and figure out what are the genes that are playing a role, what part of the genome is being changed in these different generations to figure out what those factors are.
Okay.
And sorry, you asked me one more question, right?
About the EGA specificity.
So yes, we do want to sequence. We haven't so far. We haven't sequenced the specificity or anything, but we do want to do BCR sequencing, so we can try and figure out if there's any difference there. But to be honest, we think that it is just the amount of IgA that's making a difference. That it's not really specific, because even when we have the same bugs, we're seeing different effects from having more or less IgA. So our guess is that it is the total amount of IgA, and the specificity doesn't matter. But you're right that we should test it to before we roll that out.
Thank you.
Thank you.
So Emmanuel have a question. So go ahead, Emmanuel.
Thank you. Hi, Chica. I'm Emmanuel from Mexico. I am an undergraduate student. I want to know if you have information about when there is a complementation of T-Rex population and Elios population. When you have a high amount of ROR gamma T-Rex, you have a less Elios T-Rex population, and the opposite. When you have less ROR gamma T, you have more Elios. So do you think that there is a communication between this axis of transmission of the set point establishment with the T-Rex production of TIMO? Of
T-Rex production of what, sorry? Of
thymus.
Oh, so the RR gamma Tregs are peripheral Tregs, right? So we think they're being differentiated in the gut. So we don't necessarily think that it has anything to do with higher up, like in the thymus. We think all of this is happening in the intestine. And we think it's more of having the niche availability, right? So you start off with the Helios positive Tregs. And depending on what you have, what these factors are, it's setting the microenvironment in such a way that you end up having more or less RR gamma Tregs. So if you end up having something in the niche that's going to expand your Helios positive Tregs, then your niche is sort of taken up by the Helios positive Tregs.
Okay.
Right? You see what I mean? So we think there's something that's being set in the whole environment, in the environment of the gut, that's determining whether it's Helios or whether you have high Helios or high RR gamma. Also, having said that, one thing we did mention in the paper too is that you don't have RR gamma Tregs show up in the gut until weaning. until the mice are about 3 weeks of age. Until then, it's mostly all Helios Tregs. I mean, about 90% is all Helios Tregs. It's Helios and GATA3 Tregs until then in the gut. So it's only at 3 weeks of age that this difference happens where, okay, you either get fewer Helios and more RR gamma, or Helios kind of stays up and you end up getting lower RR gamma. So we think that differentiation, like that balance between Helios and RR gamma that happens at 3 weeks is being set at 1 week. And whatever that's being set is the precursors of those cells or something in the intestinal environment that's being set. Whether it's being set in the thymus, like you asked, probably not, because for those, you know, first 3 weeks, it's all Helios Tregs in the gut, right? So whatever's happening is happening in the gut, if that makes sense.
Yeah, thank you.
Well, Dipshira, we have 2 more questions. Are you okay to answer? Yeah, okay. Is this possible that other immunological traits apart from RORY+ Treg states might be transmitted this way?
It's quite possible. So we did do extensive immunoprofiling of several immune cell types in the F1 mice. We looked at several subsets of dendritic cells, macrophages, other T cell subtypes, ILCs, and so on. We didn't really, at 6 weeks of age, we didn't really see much of a difference. We did find one thing that was different were gamma delta T cells, but we did do experiments in gamma delta PCR delta knockouts and found that there were no difference in RR gamma Treg. So we think that's unrelated to the RR gamma Treg phenotype, but there was a difference that we found in gamma delta T cells. We also did this immunoprofiling at 3 days of age. And at 3 days of age, we did find differences in ILCs. So ILC2s and ILC3s were different in the gut. And that difference, though, was not present at 6 weeks of age. One thing that we are considering is that maybe the cell type that's being set in the first week of age is maybe these ILCs. And then, you know, later on, these cells somehow control the differentiation of RR gamma Tregs. We have no evidence for that. We're just guessing that because in the first 3 days, the cell type that's changing the most in the intestine is the ILCs. And it also changes with the fostering. It's also changing in the F1 and so on. But apart from those cell types, we haven't seen anything. And in terms of non-immune cell types, we haven't looked at that. It could be that maybe the enteric neurons are changing, the endothelial cells are changing, epithelial cells are changing. We haven't explored that yet. We only looked at immune cells.
Verena asked, why do you think the time window for cross-fostering is narrower in B6 than in BALB/C? Wouldn't you expect it the other way around when the IgA present from BALB/C foster mother is important?
Yes, I absolutely agree. I was confused also when I saw the result because I would expect the opposite because BALB/C mothers give you more IgA. We're not sure why this seems to be different. One thing that could be the reason is that B6 have what we call a dominant phenotype, right? Because they're high, and BALB/Cs have a low phenotype. We're thinking that maybe once you have a dominant phenotype, it's really hard to change that high phenotype. But when you are low, when you have low RR gamma Tregs, for whatever reason, you can be manipulated to become high. So you have sort of a longer time window to do that because the mice are already starting at low. So if that makes sense. But the real answer is we don't know why. We would also expect the opposite. You're right.
And Jonathan says, hi, I work with evolutionary biology in Bern. How these mechanisms of hereditary fit with the conventional evolutionary theories?
Evolutionary theories. I don't know.
Yeah.
I really don't know. I mean, it's an interesting question though. Yeah, I'm not sure. Sorry.
I have one last question, Deepshika. In the last figure, you make this experiment where you have this valve C G change and knockout mother, and then you breastfeed a B6 pup, and then you see all this phenotype. But what would happen if you have a pup that is valve C that is knockout for this G change, and then you put it with B6 mice? So in the first days of their lives, the pup will have IgA, but at the end, when the mice is not now drinking or having milk, they wouldn't have IgA. So what would happen there?
So you're saying take the JH knockout pups and give it to B6 mothers? Yes. So we did do that actually in the paper.
Yes.
And what we find is that so when we do that, the JH knockout mice are high. They will have a high phenotype. So you only required the IgA. Oh, sorry. I see your point. No, what we did do there was, okay, no, never mind. We did not do that. You're saying give JH knockout pups to a B6 mother, not a, no, we did not do that. But
and what would you expect?
My guess is that they would have fewer RR gamma Tregs, but we don't know. We don't know. We could try that.
Okay.
Okay. And guess whole thing. Would like to thank once again Deepshika for kindly accept our invitation. We would like to know from your expert point of view, what is the take-home message of this amazing work?
So the take-home message, I mean, to be honest with you, this was pretty surprising for us too. We weren't expecting to see. The whole reason why we started this, it was actually started by a graduate student in the lab that was before me. I sort of inherited this project from her. And the reason why she was trying to do this is because RR gamma Tregs are obviously very important in colitis. So she was trying to find ways to manipulate the levels of RR gamma Tregs. And she was trying to see if in adult mice, if you can increase the levels of RR gamma Tregs, that it would be more useful for colitis. And interestingly, no matter what she was trying to do, she could never increase the levels of these RR gamma Tregs beyond what the B6 mice already had. And by doing that, she was also testing all these other strains of mice and found that all these different strains had different levels of RR gamma Tregs. And in these strains, if you gave them high inducers of RR gamma Tregs, they wouldn't change either as adults. So this is sort of how this whole thing started. And when we started doing this, I mean, it was quite surprising that they split so, it was almost like black and white. You can see that, you know, from the data. It was very, very obvious. So our take-home message is that, you know, what the mother does can have an effect basically several generations later on, which is quite exciting. It's quite interesting. I mean, this Jonathan Zeiss asked about evolutionary theories. This sort of, in a way, fits with the Lamarckian evolution, where he's saying that you develop certain traits during your life, and then you're able to pass those traits on to future generations. So yeah, the take-home message is that what you do now could have an effect several generations later, and sometimes these environmental traits that we develop and pass on can have more of an immediate effect in the whole population, faster than a genetic effect, because a genetic effect takes much longer.
Wow. So thank you once again.
Thank you. And Verena, if you have time, if you want to talk
your insight. Sorry?
Because we didn't really clarify Verena's question about the germ-free. So I told her if we had time later.
Yes.
Oh, yeah, for sure. Verena?
If you have more time, would be great. Maybe I'm just
Yeah, sure.
on the assay, but
go ahead.
I really didn't get yet where you think that this colonization.
Do you want to tell me again what
So I mean, I'm aware that the germ-free in the germ-free status don't really have raw gamete or just super low levels.
Yeah, so germ-free they're only at about 10%.
Mm-hmm. But yeah. I was still kind of wondering because as soon as you colonize them with B theta or sorry, I forgot the other back, that they kind of show this difference between Black 6 and bulb C. And when you do this short antibiotic treatment before birth, kind of they don't get back to the levels, you know, while the pups should be colonized normally. And also the foster mothers have been colonized normally all the other time, while the germ-free mothers have never been colonized normally. So
I think I'm not quite getting what you're asking, but let me try and break this down to see if this is what you're asking. So you're saying, why is it that in moms that are treated with antibiotics, with the B6 and the bulb C, why are they different in the pups? Is that your question?
So yes, why are the levels in the pups different of the gamma T cells?
Oh, I see what you're asking. Okay, so first things first, between germ-free and SPF treated with antibiotics, they're not the same, right?
Yeah.
Because antibiotics won't get rid of all of the bugs. So it's just getting rid of a majority of the bugs. But say we assume, okay, we're getting rid of most of the bugs here. The B6 pups, the SPF pups that we're giving here to the bulb C and the B6 foster mothers, they were already born to SPF mothers.
So they were colonized.
Their birth mothers had bacteria, so presumably at birth, they were colonized with some bacteria. So when we give them to the foster mothers, they already have some exposure to bugs. And what we're getting from the foster mothers is just that, what are they giving, you know, if they didn't have bugs in the last few days of pregnancy, can they still transmit, you know, these either bugs or IgA to their offspring? So the reason why we should have this also in the supplemental, I think, but the reason why these B6 mice, for example, I'm pointing again, like you can see, but in the left-hand panel, right? B6 moms that were treated with VMNA and were given bulb C pups. So there, if you look at the IgA is different from regular B6 mothers. So here what we're trying to say is, yeah, it has to do, the moms also need to have microbes in order to be able to transfer certain levels of IgA.
Which again, the germ-free mothers also don't have.
Yeah, but the germ-free is very different in the sense that their IgA is very different.
Okay.
So for example, I should have said this. So B6 germ-free mice have very little IgA compared to bulb C germ-free mice. And this has nothing to do with the microbes. Okay. This apparently is a genetic thing. There's a paper in Immunity, I think 2017. It's Franson et al. And there, if you look in their paper, they already explained that B6 germ-free and bulb C germ-free in terms of their IgA levels are very different to begin with, even though they don't have any microbes. So that has to do with some sort of genetic.
And this broad spectrum antibiotic microbiota depletion in this time window lowers than the IgA in the bulb C foster moms.
Yeah, it is changing it. We don't see because it's such a short amount of antibiotics, we don't see as striking of a difference as we normally would, which is why we think it's something also in addition to IgA. The IgA can't be the only thing changing. But yes, it does change it.
Okay, so that I think answers my question. Thank you.
Yeah, sorry I didn't understand earlier what you were trying to ask.
So
I guess that's all the questions. So thank you, Dipshika, for your insightful thoughts, and we will stay tuned of your future work for sure. And we hope you can join us in the future sessions of the Public Journal Club.
Yeah,
and
thank you.
enrich the discussion with your background. And Dipshika, do you have any problem if we take a screenshot of the session?
No, absolutely not.
Okay, so guys, turn on your cameras, please. So,
okay.
okay, so.
Yes, I like the hashtag. It's nice.
Okay, so I know we wait Guadalupe. I don't think she has.
So, okay, that's all. So thank you so much, Dipshira.
Thank you all. Thank you for having me.
Thank you.